Good morning, and thank you for joining us this morning at what promises to be a very interesting set of discussions. My name is Eduardo Macaño. I'm the Dean of Biological Sciences here at UC San Diego. This morning's discussion deals with a set of extremely important issues that affect the life of every human on Earth: biodiversity, its value, and the challenges that we face. Contributing to this discussion are 5 distinguished researchers, each with a different area of interest and each with a different perspective on the value of diversity. It is my great pleasure to introduce the first of these panelists, Dr. David Woodruff, who will present an overview and discuss new methods to preserve endangered species. David received his PhD and Doctor of Science degrees at Melbourne University in Australia. He was a postdoctoral research fellow at Harvard University with Ed Wilson and Steve Gould, teaching as an Agassiz lecturer in the area of biogeography. David is interested in animal species and their evolution and has worked on such diverse groups as Australian frogs, West Indian land snails, African parasitic worms, rainforest birds, and the living fossil nautilus. As you can see, he's truly a man of the world. Since coming to UCSD 20 years ago, he has turned his interest to various threatened and endangered species, including the rhinoceros, chimpanzees, and elephants. David has pioneered the application of new molecular genetic methods to the study and conservation of rare and endangered species. He is the founding chairman of the division's new section of Ecology, Behavior, and Evolution, teaching evolutionary biology and wildlife conservation. He also serves as a trustee of the Zoological Society of San Diego, the parent organization of our world-famous Zoo and Wildlife Park. Ladies and gentlemen, David Woodruff.
Well, thank you very much for the introduction. Thank you for coming. I hope you'll find the next couple of hours useful. We obviously believe that the topic of today's conversations is extremely important. We all take the planet's life support services for granted. The plight of biodiversity is one measure of the fact that that's a foolish strategy to carry into the future. And we will approach the problems of biodiversity conservation from different angles. Welcome to San Diego. Some of us like to think of San Diego as the cradle of conservation biology for reasons that I'll explain in a minute. I trained as an evolutionary biologist and spent half my life looking backwards trying to recreate the present. Then I came to San Diego and saw an opportunity to do what scientists are meant to do, which is to make predictions and test them, and what better field for doing that than future evolution. And so I see conservation biology as really applied evolutionary biology. And I see the challenge for geneticists and everybody else as developing the tools that will allow managers to confront what we call the biodiversity crisis, what some people call the 6th great mass extinction. I'm showing a picture of a northern spotted owl to do 2 things. First of all, I will be focusing on species, their DNA, and their future evolution. But before I do that, I want to make sure that everybody in the room understands that as conservation biologists, the first recommendation we would make to anybody who asked is forget about individual species, worry about habitat, worry about land, worry about connectivity of reserves and the like. So that's a caveat on what I'm now going to get into. Does the spotted owl have intrinsic value? It does to some people and not to others. But its critical role in this story is as an indicator species. It has legal standing. The Supreme Court of our country has determined that its value is incalculable, that is infinite, that is more than anything else. And if that's the case, then why should we be concerned? It's an indicator of abuse of natural resources. And its demise was, in fact, telling those who had the eyes and ears to listen that that was a problem. And since then, we've done something about it. So I'll be focusing on species, threatened and endangered species, but it's part of a much bigger problem. Something like 100 million species have lived throughout a few billion years of history. About 10 million are out there at the moment. We only really know about 2 million of them. And there's an enormous challenge of work to be done. By focusing on a few species, though, we can do a lot of good. And when I say that, I'm thinking that we all have to recognize that any planning, any management we do is for the survival of ourselves and these species on a planet with at least 9 billion people on it. Here's the well-known map of the United States showing the distribution by county of the number of threatened and endangered species. The intensity of the color indicates the number of taxa in different groups as diverse as fish, flowering plants, mammals, and birds and butterflies. And when this is generalized, we find that there are 3 areas of the United States that are hotspots for species in trouble: Hawaii, Florida, and the winner in this dubious contest, San Diego County. There are more listed species in San Diego County, more threatened or endangered species here than anywhere else. And so it's appropriate to have discussions like this here because we are a national test bed for responses to this crisis. How important is the San Diego story from a global point of view? We think it's important because it's 2 buildings from here in 1978 that an international meeting was held sponsored by the university and the zoo. Michael Soule was the chair of the meeting, and at that meeting the cliche conservation biology was coined. So that's why I said originally that we think that this is the cradle of what is now a new science and a new curriculum material that's spread across the world, certainly a new society, a new journal, and the like. San Diego is visible here, Point Loma and the bay at the bottom of the picture, the University of California a little bit above. The major threats to biodiversity here now are habitat destruction, habitat fragmentation, and the introduction of alien species. And we'll hear more about that in a few minutes' time from other speakers. But the San Diego pattern of what happens when 2 to 3 million people come and live on a piece of habitat is generalized around the world. We are living in the 6th great mass extinction, according to some people. Other people say, no, it's not a mass extinction. It's a crisis, if you like, but it's not truly an extinction. But I point out the bottom line on this slide that the rates of extinction ongoing across the planet today are 100 to 1,000 times background. There hasn't been an event or an episode in biodiversity's history like this for the last 65 million years. And in our lifetime, we are confronted with the dilemma of half the big things being threatened with extinction. So it's a global problem writ large here in San Diego and in Southern California. Now I'm going to focus on my particular side of this story. I'm a conservation geneticist now, according to some people. What do? We define units for management, be they species or populations of related individuals. We establish the historical patterns and processes that gave rise to those taxa, so that managers might mimic or emulate them in the future. We can, and we will increasingly have to try to find out how much variability is left in these populations and do everything we can to maintain that innate variability, because variability is correlated with fitness in an evolutionary sense, with ability to prosper under conditions of changing global climate, with the ability to resist the appearance of emergent diseases, with the ability to resist the stresses that humans put on biodiversity. And geneticists can, of course, do other things. How do you do genetics of conservation? It's simple. You get a blood sample, you do some alchemy, and at the bottom of the slide, we get some DNA, which we run out on gels, and I don't need to tell you what it looks like. So my first challenge when I came to San Diego was to do a paternity case in rhinos, and we went out and and do you know how hard it is to get a blood sample from a mother rhinoceros? There was simply no future in doing this type of wildlife genetics in the early 1980s. And it wasn't until late in the 80s that a breakthrough in another field was co-opted into the service of biodiversity conservation. And here's a student of mine at an orphanage in northern Thailand demonstrating the two-handed DNA sampling method. He's simply stroking these animals, and from the hair samples that fall out into his hand, if he's clean, we can eventually get gibbon DNA, and from that we can do anything we want in the world of genotyping. So I've got 4 examples to tell you about that exploit the PCR reaction and the ability to genotype animals non-invasively, non-intrusively, and without ever seeing them. First example involves our closest relatives, the chimpanzees. And we went to Africa and we got hair from chimpanzees without ever touching them, and we were able to answer 4 questions. We were able to establish their real pedigrees and get into the details or the formal genetics of their love lives. We were able to show how much they used to move around before their habitat became fragmented. And most important, we found there's not one chimpanzee, but 4 separate chimpanzees. Call them subspecies or species, your judgment, but managers have to manage them from now on as separate taxa. The second example involves a bird, and so obviously it involves feathers. This is a San Clemente Island loggerhead shrike. From a single breast feather we can get enough DNA to do the genetic reconstruction of the history of this population, of this taxa, and we can talk about its future evolvability. We made 3 discoveries. First of all, the island population is genetically isolated from look-alike birds on the mainland. Secondly, it's been genetically isolated for 100 years. We went back to museum drawers and got DNA out of feathers of birds shot in about 100 years ago. And third, we found out that in that 100-year period, the island population has lost variability. It's isolated. And so there is a genetic time bomb ticking in that population that could eventually lead to its extinction through genetic disease or chance. So hair and feathers. How endangered is this bird? There were 14 left on the planet when I got involved. Now, as a result of captive propagation, there are more, but it's certainly still in the critical phase of its survival. The third example involves elephants. Hard to get hair, no feathers, we stooped to poop. And so a student of mine went across Africa and discovered that there's not one elephant in Africa, as the books tell us, but there are 3 elephants. There's a savanna elephant, there's a forest elephant, and there's a West African elephant, and that result isn't even published yet. But managers from now on are going to have to thank us for making their task much harder. In return for that grief, we have done something of great use to managers. We've shown them how they can census a dangerous, sometimes unobservable forest elephant population. We went to a tiny park that's isolated near the coast of Ghana in West Africa, and we walked transects through that park over a series of a few days, and we collected DNA from dung samples, and we established that there were 69 adults and 17 juveniles in this period of time, and we know the sex ratio precisely because we can sex the dung, and we know from the capture rate of new genotypes that there are probably about 212 elephants in that park. But nobody's ever seen them, and nobody would ever have the time or live long enough, they'd be killed by the elephants, to go out there and census them the old-fashioned way. So we've now got a molecular genetic way of censusing hard-to-find animals. And that's going to be useful here in this country, and it's already being used in various areas, for example, in Yellowstone wolves that you might hear about later this morning. The last example of the use of molecular genetics in the service of the biodiversity crisis involves parks like that one in Kenya where you have something left inside that you are trying to save. The process of fragmentation is global. It's here in San Diego, it's in Africa, and I'll now show you an example from Thailand. When you fragment a large population, you create small populations, and the process of evolution speeds up and enters what we call the vortices of extinction. 2 things happen on the isolated fragments. First, species disappear by chance or competition. And second, the individuals that survive in the populations that manage to survive start to erode from a genetic point of view. Through inbreeding and genetic drift, they lose genetic variability and they become more susceptible to extinction by normal evolutionary processes. Now, we've known that curve on the right. We've known about it in theory for 100 years, but we've never had genetic markers sensitive enough to monitor the process. And now I'm going to show you the last story of work done here at UCSD, showing how you can develop a method to monitor the collapse of populations. In the south of Thailand, the government flooded a large valley and left in the reservoir behind the dam wall 100 small islands. And on those islands we were able to collect, to live trap small mammals, and from the small mammals we were able to take hair samples and then release the animals and go back a year later and a year later. And over a period of 8 years after fragmentation, the yellow line there shows the collapse of the population relative to, at the top of the diagram, the top of the picture, a red dot indicating the mainland control. So in a period of 8 years, a significant loss of genetic variability in a mouse and in a tree shrew, the red line indicating the control mainland population. So now we've given managers a chance to monitor a process we've always worried about but ignored because we couldn't measure it. I don't care about saving the rats and mice of the forests of Thailand, but I am interested in whether their national parks will save the big things that they believe they will. The answer is they won't. They won't because they're not large enough, there are no corridors between them, and so eventually the animals that we're trying to save in those parks and in parks in this country are doomed to genetic erosion, and that will complicate or exacerbate their management. There is a way around it, and that managers don't want to hear about because it involves more work. You have to put the elephant on a truck and take it around for visitation once a generation, and drive it around between parks to artificially reinvigorate the gene pool locally. And I've termed that new way of managing nature bioneering. Whether that word catches on, we don't know yet, but we have a lot to do if we want national parks to do what the public expects them to do. 3 quick examples of bioneering that you'll hear more and more about in the next few years. Local examples. California Condor Recovery Program involves the selection of individuals and their behavioral training for release into the wild. Bioneering. Second example involves artificial insemination and the production of giant pandas here at San Diego. Intensive medical procedures used to assist reproduction, and we'll see that more and more to deal with the basket cases of the biodiversity crisis. And the third example involves cloning, and this is Kurt Benerschker at the Frozen Zoo he founded. 20 years from now, it is conceivable that science will have advanced to the point that cell lines can be woken up and can be used to grow up new individual animals. Now, we're not talking about saving animals. We're talking about species that have already gone. And there is absolutely no hope of ever reintroducing them any other way. Too expensive to contemplate today, too fanciful to claim we can do it today for the species that need help, but it's certainly something that some of you are watching and will be on the news in increasing frequency over the next decade. The third thing to watch is changes in our approach to biodiversity conservation, and we're seeing that here in San Diego County. For the first time, different government groups have come together with landowners, would you believe, with the NGOs and the animal lovers, would you believe, to plan the future development of the county. Will it work? We don't know. Is it worth the effort? Absolutely, because it's the only way to go in the future. Here at the University of San Diego, we have mounted a small program in training what we hope will be the next generation of scientists and educated members of the citizenry in the problems and how we might approach them. I'd be happy to talk to you individually or take questions later. Thank you very much. The next speaker is Professor Martin Crispiels. He is a member of the Division of Biology, a member of a growing cohort of scientists who are addressing the human need for more and better food. So he represents an entirely different stance on the biodiversity crisis, and we'll talk to you about food and agriculture and the latest scientific developments in that area. He's a member of the National Academy of Sciences and a distinguished plant geneticist in his own right. Martin Crispils.
Well, I want to tell you a slightly different story, that is something about the agricultural connection to biodiversity. The first message is that agriculture is the biggest driver of environmental change and degradation. Now, one could say that that's a sort of necessity. That is, you could also say that it's people. It's the tremendous population explosion of the 20th century, so that we now have 6 billion people to feed, and that has caused enormous natural ecosystems to be converted to agricultural ecosystems. So agricultural ecosystems like these look beautiful. You go to Tuscany and you see the vineyards, or you go to Spain and you see the olive groves, or to the Napa Valley, and you say, wow, it's beautiful here. And it is. But they are nevertheless agricultural ecosystems that, in terms of species diversity, are much poorer than the natural ecosystems. So it's not just the conversion of the ecosystems, it's also the use of chemicals, of pesticides, of fertilizers, etc., that has an enormous impact on the world. All that to feed the people. Now, we expect another 3 billion people by the year 2050. And so that means that we will have to double food production again. If you look at the causes of the loss of biodiversity, then they are, as I said, the conversion of natural to agricultural ecosystems. And here you see a map of the world, and in color are in all the cultivated land, all the arable land that is now being cultivated. Now, if you look at that map, you could say, well, there is still a lot of room there. But if you look a little closer, you see that much of that, the areas which are in faint yellow, are actually the deserts or the very cold areas of the world. So basically, we have used already all the arable land that is available, with the exception perhaps of some land in the center of Africa and some land here in South America. Nearly all the arable land has been used. Now, the other aspect of the loss of biodiversity that is the result of agriculture is that we have replaced, or we are replacing, populations with greater diversity with populations of much less diversity. Now, here you see a domesticated tomato surrounded by 6 different wild tomatoes. So the wild tomato varieties that are growing in the Andes are actually very small tomato fruits that are usually green. A few of them are red, and you are well familiar with our domesticated tomato. Now, because of the expansion of the agricultural area, it's again an issue of habitat destruction. That is, the habitat for these wild species of tomatoes is rapidly disappearing. Now, or has disappeared already. Now, and with it, of course, the plants, and with it, the genes that they contain. Now, during the domestication of agricultural crops, what evolved were what agronomists call land races. So land races have a great genetic diversity, as shown here in this picture of corn cobs. The result of plant breeding efforts is that plant breeding companies started to release elite lines, which of course produce much more abundant crop per land area, per acre, but are much more genetically homogeneous. They produce more because they have been highly selected. But the result is that all the genes that are present in these land races are being lost, because farmers happily switch, at least if they have the funds to buy the elite lines, they happily switch to elite lines because they produce more crop. So that in a way is also the result of the need to feed more people. Now, having elite lines with a narrower genetic base can have negative consequences. As shown in the United States in the 70s, we had a disease called southern corn blight that ravaged the corn crop in the southern tier of the US and caused something like a 20% loss of the corn crop in those states. Now that was due to a sort of coming together of a certain weather pattern together with the fact that much of the corn had a gene that made it susceptible to this particular disease. Now that was due to the fact that we had that elite lines were being released, but unbeknownst to the plant breeders, all these elite lines had this particular susceptibility gene. So that's the negative aspect of narrowing the genetic base. Now, this problem was quickly solved by the plant breeders, and now this gene has been eliminated from the corn crop. So how can molecular agriculture help in preserving diversity? First of all, we need to enhance agricultural productivity. That is, if we have to double the food supply and there isn't much more arable land available, then it stands to reason that we have to double the productivity per acre. Otherwise, we will have to put even more land to the plow, so to speak, and as Dr. Woodruff explained, that will cause even greater loss and greater fragmentation of whatever natural areas are left. Secondly, we must characterize and conserve genetic resources. That is, the wild relatives of our crop plants and the land races have to be characterized, their genes identified, their important genes identified, and they have to be preserved, and I'll show you that in a minute. And finally, we have to decrease the environmental impact by creating novel crop varieties. Now this goes back here, that is, only by having novel crop varieties can we also increase productivity, and to create novel crop varieties we have to conserve the genetic resources. So here you see that this is the same map except blown up a little bit. You see that the different colors refer to the productivity of the land. You see that some land, for example in Europe, is very highly productive. That means in terms of pounds or dollars per acre. Other land is moderately productive, and a lot of the land, especially say in Africa, is still very poorly productive. So there is a lot of scope to increase the productivity of the land we have, but that will require research and development of these novel crop varieties. Secondly, here you see what is being done for preservation. So people have seed banks. These are huge cold rooms. This is one of the USDA, which is in Colorado, where seeds are collected worldwide and are stored in these cold rooms or freezers, huge freezers really, and so they can then be preserved for later times and for use for planting out, for assessing the particular properties of those plants. So this is where both the wild relatives and the land races are stored. However, this is not possible with all plants, and here on the right you see a tissue culture facility. So we have many plants that don't propagate by seeds normally, such as potatoes, for example. So those have to be stored as cuttings in tissue culture rooms, and those have to be transplanted, however, they have to be under sterile conditions transplanted, let's say once every 2 months, every 3 months. So this is much more expensive to maintain these kinds of species for posterity than it is to preserve species that propagate by seeds. So, as I have indicated, there are two aspects to this. That is, landraces are domesticated varieties that are genetically heterogeneous, and they are adapted to local climates. So they contain gene combinations that are very important for local areas. And they have genes that we may want later. So that is the reason why they have to be conserved. The wild relatives also have to be conserved for the same reason. That is, they contain genes that we don't yet know about. So this is almost too much genetics, but you see, what makes you unique is not your genes, as is always said. What makes a plant unique is not its genes, but rather it's the alleles. It's your particular copy of that gene. That is what makes you unique, and that is what makes the plants unique. So if we conserve landraces and the wild relatives, then we conserve those particular alleles, and they later may prove to be crucial. So making new gene combinations, so now where does molecular agriculture come in? Making new gene combinations and novel crops can take two routes. First of all, we can simply stick with traditional crossings and selections. However, the science of genomics will greatly accelerate our ability to make these kinds of crosses and do these kinds of selections. Secondly, we can do genetic engineering, where we switch genes, or we introduce genes from any organism into a crop plant. This is some of our own work, where we took a bean gene and put this bean gene into peas to make them insect resistant. So these are the controls. They are, every hole here represents an insect that has burrowed into the seed, and this is the genetically engineered population, which has this bean gene expressed, a bean protein actually, expressed in the seeds, and is now insect resistant. So these are novel crops, you could say, novel gene combinations. Now, if we now go back, we have to preserve all those land races, all those wild relatives, because they might contain genes that can be used in these kinds of applications. So this is then my last slide. So what is the role of molecular agriculture in preserving biodiversity? It's to create new gene combinations for crop plants by using the genes from many different organisms, with the goal of increasing crop productivity and preserving natural ecosystems. So those two go always hand in hand. So we need to preserve the genes if 50 years from now we're going to be able to use them to create the crop varieties that will be needed. Thank you.
The next speaker is Robert Fisher. Robert graduated from UC Davis and was a postdoctoral scholar here at the University of California, San Diego, with Professor Ted Case and his group. He's now an adjunct faculty member in the biology department at San Diego University. He's employed as a research zoologist by the USGS here in San Diego. Some of you may know or have his handbook to the reptiles and amphibians of Southern California, and he's going to talk about endemic San Diego fauna, what's being lost, why should we care?
Thank you, David. I'm actually, I'll talk a little bit about this, but I'm going to follow through on some of the stuff that Dave talked about, biodiversity in general, but with a more kind of local flavor to it. So just to put ourselves in perspective, everybody's pretty familiar with this. This is the biodiversity hotspots globally map of them, and you can see that the South Coast ecoregion and the California floristic province always comes out as a significant place globally, and so there's global significance to our region locally, and I want to just, I always like to frame that. And this recent estimate is that an investment of $6 billion would protect the highest priority regions of the hotspots globally and the majority of tropical wilderness areas, and that would encompass an area just about the size of Alaska. And so globally it's not a large investment to potentially protect the major hotbeds of evolution in the world. On each of my slides, I've tried to incorporate a reference for these so you can get more information from those websites as well. And so I wanted to kind of give you the information and then let you follow up on it if you need to. So I want to talk about framing our area, the floristic province, and then some of the work that we started in 1995 when I was a postdoc here at UCSD was looking at kind of the land use planning that was taking place in Southern California and understanding kind of biodiversity at this local scale within this floristic province. And this is a figure out of a recent paper that myself and Dr. Ted Case and a former grad student here, Andy Suarez, had in conservation biology. And this is Los Angeles, here's San Diego, and what we did was we set up a series of sites across this landscape to try to assess what's going on with reptiles and amphibian diversity. And then since I've been in USGS, we've kind of expanded on some of these sites and are looking more at other diversity elements as well, looking at mammals and ants and other species. And the core of this project was by using the same design, we used these pitfall traps with drift fences nested within sites and then across regions, we could start assessing biodiversity across this region and look at the effects of fragmentation, effects of urbanization on this diversity. And so these are just some statistics from some of the work we've done. And again, this has been taking place for the last 7 years. And then by using the same study design, by using these buckets and fences and snake traps, we're able to actually compare our sampling across these regions, across counties. And now we've expanded since then to where we've got sites that are as far as Santa Monica Mountains. Some areas in the Bay Area, people are using our same techniques, so we can actually compare fragmentation in the Bay Area to fragmentation in Southern California to see if the same species are responding in the same ways. And some of the results from Southern California include, we've recorded 46 species of reptiles and amphibians. A lot of this information and pictures are in the book that we put together in '97, and then also on our website through USGS we have an online field guide version, so you can go and see pictures of the species and then get descriptions of them. And we've got over 60,000 captures of reptiles and amphibians, and sites vary from having 9 to 33 species present, and some of the sites that have less diversity are the sites that are more highly fragmented in more urbanized matrices, and the sites with higher diversity are the sites that are more intact. And we've been trying to assess which species are more susceptible to local extirpation and extinction. And then at individual points, we might have up to 22 species of reptiles and amphibian present. So we're getting a handle at the local scale, at the point, what diversity looks like then within a site and then within this whole region, and how different species communities are developed, and then how they're responding to urbanization, fragmentation. And so what we can do is take some of this data then and we can visualize differences across species. And so what we've done is using a kriging technique in a GIS application, and we've looked at 2 different species. One is the horned lizard, and this is San Diego and LA again. And then by taking our capture rates at different sites, we can look at how this species' relative abundance varies across space, across this local area at Southern California, and we can compare that to the western fence lizard. And we can see that there's these darker colors are higher density populations. So the horned lizards really like sandy beach dune habitats, which only persist still at Tijuana Estuary, and they also like some chaparral habitats, whereas the fence lizards prefer a lot more edgy habitats. They don't do so bad with urbanization, and they really don't like areas that are hotter and drier in Riverside County. And so by visualizing how different species are dealing with this, both this urban matrix and then also the environmental variables locally, we can get a better perspective on how these communities have developed and then how they're responding again to fragmentation. And I'm going to come back to the Scoloporus and to the fence lizard in a few minutes with regards to its ecological importance and significance locally in Lyme disease. So one thing we've been able to do then is document declines in snakes across this region. This is data for San Diego County for some of our sites, and these sites in green are what we would call core areas that are not fragmented yet. And these sites that are in red are fragmented areas, such as Point Loma and Tijuana Estuary. And what we've got here are the dark blocks are sites where we've recorded snakes within the last 7 years at these individual species. And then these crossbars are sites that we have records from the 20s and 30s for snakes at those sites that they're now absent as far as our data is showing. So we've got a set of snakes that is declining from these fragmented landscapes. And so our hope through a lot of the research we're doing is to identify what are the factors causing those declines so that we can limit those declines as these other core areas become fragmented with increased urbanization. And then two factors that stand out are the impacts of roads and the impacts of light pollution, which are secondary impacts of urbanization, obviously. And so if we look at this change in road, which is a risk factor for diversity, we see that in 1938, these are vehicle miles driven per day in San Diego County. And we can see that there was very few vehicle miles driven per day. It was, I think, 2 million miles per day in San Diego County in the 1930s. And there was some statistics done at this point by Lawrence Calaber that indicated that this resulted in about 10,000 dead snakes a year in the county. And then these are data from Sandag showing the levels that we had for vehicle miles in San Diego in the mid-90s, and then the projection for the next 15 years or so. And so you can see that as a risk factor, even though we'll have these reserves, and a lot of them have hard lines and they're protected, we still have risk factors within them and associated with them. Another is light pollution, which is, here's a picture from Mount Wilson of Los Angeles in 1908. And then here's a picture of the same picture in 80 years later. And you can see this urban light pollution bubble. And this is from, I didn't put this on here, but it's from the International Dark Sky Association, their website. And this affects circadian rhythms, obviously, but it also affects a lot of nocturnal species activities, because in a lot of instances in these cities, you're washing out the lunar cycle. So any species that are tracking a lunar cycle are now not going to see that. And it's going to be actually, in a lot of cases, more bright all the time than it is during the new moon periods. And so we think that's a factor locally in declines in some nocturnal species of snakes. And then we were also doing some genetics across this region using the same techniques that Dr. Woodruff discussed. And this is one snake that we've looked at in detail. I've got a graduate student that's working on this. This is a red racer, and I actually brought one we can look at later. And what we found was when we started our work, there was one species that goes across the country from the Pacific Ocean to the Atlantic Ocean. And what it looks like, the data we're finding, is that we actually have maybe 3 different species in San Diego itself. And so here we've got one species, we thought, nationally, and now it looks like it may be 3 species locally. And the biggest difference is between these Baja California animals, which are right along the border zone, within about 15 miles of the border, and then the animals a little bit further north, like right here at the Elliott Reserve, which is a reserve that UC San Diego has. And up to Camp Pendleton, we have a big genetic break between these. And then we have an LA Basin form that we discovered, which is a surprise, potentially discover a 6-foot-long snake in the LA Basin that's genetically distinct and might need its own type of management. So it's taking the work that, like Dave's talking about, with elephants across Africa, and then bringing it down at a local perspective and saying, look, we have to, even at the reserve level, we have multiple different species we have to manage for. So in some areas, red racers might be doing fine, but they might be a different species than they are in Riverside County or somewhere else. And so we have to think, once we kind of understand these patterns of evolution, we can then link that back to our management on the ground. Some of the things that are very interesting to me, and I've been trying to track this, is that we've got these amazing new species that are discovered under our nose. This is a proxy for this is a sea bass related to one that was just discovered in 1995 off of Scripps. And so this is this paper here is a reference for it. It was a new sea bass. Sea basses are commercially important species, obviously, and yet we can discover new species right off of Scripps. And so if we can discover new fish right off the coast here, we have no idea of kind of what might be in our terrestrial habitats and in areas we haven't really looked. And to step back in perspective, this is a new shark species that's 14 foot long that was discovered in 1976 off of Hawaii. This is the megamouth shark, and this is now a map of its known distribution worldwide. And what you can see is the second specimen ever collected was off of Catalina in 1984. 14 foot long shark collected off of Catalina, and then the sixth specimen is this one here, which was collected live off of Dana Point. And so there's only 16 individuals known of this species, but they were never discovered until 1976. This one was actually radio tracked off the coast of California, off of Dana Point in 1990. And that was the sixth individual ever known. So with regards to preserving and conserving biodiversity, we have laws that protect specific species we know. But there's a lot of species we don't know about. And so we have to be able to have a vision to protect these habitats and landscapes, like Dr. Woodruff was referring to, for species we don't even know exist yet. And then there's a lot when you get down to the more micro scale and we start looking at invertebrates and stuff. We have a lot of undescribed and unstudied species that we're now discovering from our data and from our study sites in Southern California that may go extinct before we even get names on them. And so this is a silk spinning cricket. It's called Nematetics. And this genus wasn't even known from San Diego before the work of myself and Dr. Case. And this thing actually spins silk in tubes with silk it produces in its mandibles. Silk is obviously a commercially important commodity, and yet we have a local cricket that's creating silk that wasn't even known from this county, and maybe a new species. And then I brought some live Jerusalem crickets because there's some significant findings with Jerusalem crickets, which have a lot of ecological roles. They are hosts to parasites like horsehair worms. They're food of a variety of megafauna like coyotes. And so, yeah, most of those species haven't ever been described. And then I want to talk a little bit of the point of this is talking about the value, putting a dollar amount on biodiversity. What's the value of biodiversity to society? And so here's the fence lizard, which I showed you, visualized kind of its distribution across this region. And recent research by Dr. Robert Lane up at Berkeley has shown its importance and the significance of this lizard to moderating Lyme disease in California and on the West Coast. And so what happens on the East Coast is that where Lyme disease is much more significant an issue is that Lyme disease, the Borrelia, uses both mice and birds. And in the West Coast, it's also using these lizards. And these lizards have a borocidal compound in their blood that knocks out the spirochetes in the ticks before they end up getting on to humans. And so this professor from New York University estimates that Lyme disease costs society about $1 billion per year. That's a value to society, $1 billion per year from this disease. If we didn't have these lizards in California, that cost would be much higher. Here's some statistics, a little bit more background on Lyme disease. It's this Borrelia. This is what it looks like, a spirochete. It was first discovered in 1982. This disease that costs society $1 billion a year was first discovered in 1982. And it was thought that there was one strain. Since then, there's about 100 strains in the US, 300 worldwide. And then you can see these are the numbers of reported cases of Lyme disease per year in the United States over time. And so there's been this increase since '82 when it was first discovered. And I'll show you a map of the country. And what you can see is these are the areas that are, this is from the CDC website. the Centers for Disease Control, and these are the areas at risk in the country for Lyme disease, and the whole West Coast is at risk, including most of California. Yet, because of the boricidal compounds in this lizard's blood, we didn't even have any idea that it was playing this role in the environment to protect us from Lyme disease and knock Lyme disease out. And you can see these are the Lyme disease cases reported by state with cumulative totals, and New York has 32% of all cases of Lyme disease ever reported, whereas California ranks 10 with only 1.5% of all Lyme disease cases ever reported. And that would be much higher if, like I said, we didn't have these lizards. And these cases are because those are incidents of the Borrelia going to both mice or birds and not going through the lizard as an intermediate host. These are real numbers of cases, and that's estimated to being 10. There's a factor of 10 and how many real cases there probably are. These are verified cases, but the estimate is that there's 1.5 to 2 million cases of Lyme disease in the U.S. and not 200,000 since 1982. And so I'm going to end with this. So biodiversity is at risk both here and now. And there's many unknowns, unknown species awaiting discovery, and unknown ecosystem functions. And then there's a variety of values to humans and society for this biodiversity that we don't know yet, and it's going to take a long time to understand. Thanks.
The next speaker is Ethan Beer, who's a professor in the Division of Biology at UCSD. He received his degree from Harvard University. He's received numerous awards from the Sloan Foundation, McKnight Award in Neuroscience, Basil O'Connor Award from the American Cancer Society, and he is by trade a developmental biologist. He works on the early neural induction, which is a common phenomenon or shared phenomena in both vertebrates and insects. He's also using fruit flies to study the basis of human diseases. So here is an individual who we don't know if he wears boots or not, or whether he goes out to save wildlife, but he has a lot to bring to the table, and we owe, I think, a debt to him for being one of the co-organizers of this particular gathering, reflecting, I think, the fact that scientists from all persuasions and all disciplines have a great deal to offer the challenge of dealing with the biodiversity crisis. Ethan is going to talk about the future of human genetic diversity in the age of the genome. Thank you.
Thank you very much, David. So yeah, I'm going to give a slightly different angle on this, and it's a little bit less immediate than the very important discussions that we've heard that I think are the classic sense of what biodiversity means, the diversity of organisms in our environment and the tragic loss, imminent loss that, that we must avoid, I believe, at all costs. But I'd like now to have you think a little bit about the very near-term future with respect to human beings themselves and the potential risk that we may be at if we don't think of the issues and lessons that we've learned from biodiversity in the field, both at the level of wild species and at the level of agriculture. If we don't learn lessons from this, what risk we might be facing in terms of human genetic diversity itself and the tragic and perhaps catastrophic consequences that that potentially could have if we ignore the problem before it strikes. This is perhaps the most famous patient suffering from a disease, a connective tissue disorder called Marfan syndrome. It's manifested in part by the long, gaunt appearance that Abraham Lincoln had. And in many instances, in middle age, it can lead to severe medical complications, coronary failure, and death. So it's a significant disease. And it was recently there was a case in which there were two parents that were carriers for Marfan disease, and they wished, by using the sort of modern technology of in vitro fertilization, to avoid having their kid grow up with this disease. Although, again, one has to see right off the bat that this is a disease that did lead to one of the more productive contributors to our nation's history. So one has to be cautious about deciding whether a disease really needs to be eliminated or not. But in any case, there are serious medical consequences of Marfan syndrome, so I think if I had been in their position, I probably would have come to the same conclusion. So a carrier is a situation where a person has one good copy of a gene and one defective copy of a gene, or mutant form of a gene. And again, when I talk about a defective or mutant copy, that has to be said in a very qualified way. It really is just a variant of the gene, as Martin pointed out, an allele of a gene, a variation of a gene. And in some cases, those alleles, those variations can cause disease, but one should be cautious about calling them bad without qualification. But in any case, here's a situation where there's one normal copy of the gene, one variant that causes Marfan syndrome in both the mother and the father. And they were concerned that their kid would grow up to be Abraham Lincoln. And so what they did is they did in vitro fertilization. They generated many embryos, 8 embryos from this. And then they had them genotyped to see what their genetic makeup was. And there are 4 possibilities to what they could be. They could have gotten a good copy from mom, a good copy from dad. They could, like the parents, get a good copy and a bad copy so that they have one good and one bad, or they could have 2 mutant copies of the gene so that those offspring embryos could potentially, would in fact, have Marfan syndrome. And so what this couple opted to do is to select the 3 out of the 8 events that gave rise to an absolutely clean copy. That is, their kids had no bad copies of the disease, so they weren't like the parents. That is, they were absolutely free of the possibility of delivering the same genetic aberration to their offspring. And this quarter of the progeny, which is again sort of eugenically cleansed, has been cleansed from this mutation, is normal in terms of their life. But so were the carrier offspring that were embryos which were discarded, along with the 1/4 of the embryos that would potentially have Marfan syndrome. But these were really the only embryos with the sad face here that could potentially, that would be ones that you would seriously want your children not to be. If your children happen to be carriers, well, they'd be no worse off than you. You'd be a normal human being and could go off and lead perfectly productive lives. And the gist of what I'd like to leave you with today is that although on a scale like this, where we're looking at only a single gene, this is fine. I don't think this is going to cause any major crisis in the human genome pool. But if one generalizes this as a strategy to the future, where we will be able to look for many, many more genetic variations at a time and perhaps impose what I would sort of see as this draconian measure, which is to not only eliminate the homozygous or double mutant affected group, but get rid of the carriers as well. In this method, in this sort of approach, then I think we could be at great risk for losing genetic diversity. And my recommendation is that if one performs such tests in the future, that the outcome be simply to eliminate this category and to be blind to whether they are the purple or the blue category, whether they are carriers or absolutely cleansed of the disease. And I think if one takes that very simple, commonsensical approach by just eliminating the ones that you know are going to have a serious medical problem, then that will make that will greatly minimize the risks that what we could otherwise face. So let's look at then the problem in terms of the genome. Just basic few pieces of information here. A gene, as probably most of you know, is the basic unit of inheritance. Genes are strung together in a linear string along chromosomes that typically consist of 1 to 5,000 genes. A genome such as a human with 23 pairs of chromosomes is believed to have 30 to 50,000 genes. And genetics in the medical profession has come up with an estimate that of those 30 to 50,000 genes, there are about 5,000 of those which, if altered or mutated, can give rise to a genetically transmissible disease. So if we're considering now the sort of Marfan idea, but now on a genome scale, what you could imagine could happen, and I think is in fact very likely to be the reality within a decade, is that the parents who are thinking of having a child will have their DNA genotyped. And it's believed, it's already in the commercial sector possible to genotype all of the genes that exist in a person at a rate of overnight. with that short degree of time. So there's, I think, very clearly going to be the expectation that within a decade or so that we all have our genetic material known, at least the regions that are important for function in biological processes, for every human being. And so, and this may even be done someday at birth. So as soon as the child is born, a little bit of blood would be taken, or hair taken, and their genetic identity would be determined. Now, based on that, you will find that it's, and this is generally known from populations, that the average human being carries on the order of 10 to 20 mutations in them, variations, which, if you had 2 bad copies of them, would cause disease. And that's part of the reasons why it's not a terribly brilliant idea to intermarry with your cousin and so forth. And if you look at some of the old lineages of kings, one of the problems that led to such a high degree of cretinism was, in fact, this high degree of interbreeding, which, as Dr. Woodruff has pointed out, can in fact lead to the ultimate demise of species when the numbers and variation, genetic variation, are reduced. So you imagine that there's a mom that carries some genetic risk as a function of these mutations. She's a carrier for these, so for each one of these genes, she's got a good copy too. But these are her bad copies of these different genes. And the father similarly also, he's a carrier for all these different genes. He has a good copy and a bad copy of each of these. And if you ask, well, which ones of them do they have in common? Well, it turns out to be a subset. In this case, I've chosen 3 of them that they have in common. So now you can imagine what I would call a conservative sort of use of genetic testing versus an aggressive use of genetic testing. The aggressive use of genetic testing would be say, hey, let's clean these babies out. We want these kids to be clean. No genetic liabilities of any kind at all. So what do? Well, you find all the genes that are possible, genetic variants that are possible problems in the mom, and all the genetic variants that are possible problems in the dad. And now we do a sort of massive scale in vitro fertilization that involves maybe 10,000 embryos. And now we have all these different possible combinations. And the only one we want, in this sort of extreme example, is the good one that is completely clean. It's not a carrier for any bad thing. Okay? And that's just this one little happy tot right here. Now, the alternative, which is what I would propose, is instead of taking that very draconian method, is to say, there's nothing wrong with any of these others except this one. This is the only one that you really need to take out of the population. So why don't we do the test in that more conservative way and use that test in the more conservative way, which is only to remove this one from the pool, or the one that could have 2 bad copies of this gene or 2 bad copies of this gene. But let's not worry about those that are carriers. Let's just leave the carrier population alone and treat it just as equal to those that are absolutely, in parentheses, cleansed in this sort of stringent eugenic kind of approach. So why? Why worry about that? Why is that such an important thing to do? What's the risk of taking out these bad mutations that could cause disease? Well, here's perhaps the most famous example of why not, which is the case of sickle cell anemia. Sickle cell anemia is a disease that affects red blood cells. If you have 2 bad copies of this gene, it's pretty bad. Many children die before the age of 2 if they have it. Their red blood cells are highly abnormal in shape, their function is greatly reduced, and they die horrible, asphyxiating deaths. So I'm not going to question the judgment of a parent who wouldn't want to have their child have 2 bad copies of sickle cell anemia. So now if we sort of reconsider the case that we had initially with the Marfan example, where we have a mother that's a carrier for sickle cell anemia as well as a father, then what you have here is a situation where, again, the 1/4 that would have the 2 bad copies in this case is seriously affected, much more severe than Marfan syndrome. But here's where the subtlety and important distinction needs to be made, which is that the parents, which are carriers, give, again, half of their offspring, if one doesn't tamper with it, would be, like themselves, carriers. And that particular half is resistant to malaria. So the cleansed group, they're happy in the sense that they're not sickle cell, but they're not happy if they happen to be around mosquitoes and in a place where there's a high prevalence of malaria. Whereas the carriers, which are also normal, in their appearance because they're simply carrying a bad gene are in fact for reasons that aren't completely well understood, but due to the fact that the shape of the red blood cells is not exactly like a normal red blood cell, they are resistant to the malarial parasite, which depends critically on the shape of the red blood cell. And so in this case, one would certainly only want, if you were living in a region of mosquitoes, only to select against those that had 2 bad copies of the sickle cell gene. Now, this is an example that's been touted in the textbooks for years, and you might think, well, this is just a really rare example. But in fact, it is now believed that there are many diseases, in fact, perhaps all of the diseases that are very common, may in fact be examples like this. The reason that they're common is that under some circumstances, they're really not bad, at least in one copy. They're just a variant. And that variant, under certain circumstances, can have some positive selective advantage, which is why they're in the population. So if you just start getting rid of genes because when you have 2 bad copies, they're bad, you might be missing many situations like this, and where, in a sense, when they have 1 bad copy, they're just a variant that has an advantage. And you certainly don't want to get rid of that trait, even though it is linked in the homozygous or double mutant condition to a disease. You want to keep it around. You just want to make sure, with genetic testing, that the child doesn't have 2 bad copies. That's a simple, conservative way of using genetic testing. So I'd like to end by talking about, well, what other issues can there be besides the fact that, you know, some of these genes like sickle cell anemia and maybe some other disease genes that are common might actually have a beneficial role by themselves. So resistance for malaria, for example, in the case of sickle cell anemia, is an example of a good thing that might be associated with the disease gene, in this case with the disease gene itself. But then there's probably a far more important impact of genetic screening, of promiscuous genetic screening, which is that if this is, say, a bad gene that is going to be involved in cystic fibrosis, say, and you don't want your child to have cystic fibrosis. And now we're looking at a person that's a carrier, that's got 1 good copy and 1 bad copy. And so what we're going to do is we're going to say we want to get rid of this gene. Well, when you do genetic testing, you're looking at the genetic types of the different embryos. You're going to basically not just get rid of this gene, but you're going to get rid of all of the other genes that are right next to it, linked to it on the chromosome, because that's how the genetic testing is done. Whole chromosomes get given to the offspring during reproduction. So if you get rid of this gene, you're also going to be getting rid of, at very, very high frequency, genes that are nearby. The closer the gene, the more tightly linked it is, the more often it will be gotten rid of along with the bad gene. So this gene, for example, could be a gene that confers, as is known in terms of hypertension, a genetic component to lowered blood pressure. Wouldn't necessarily want to get rid of that. Here's a really interesting one, which is immunity to plague. I'll get back to this one in a moment, but there's been a speculation that there is a genetic variant of a gene that, when it's in a mutant form, has perhaps made people resistant to the plague back during the European Black Death epidemic. And there are other things. There's a strong genetic component to longevity. You wouldn't want to get rid of a good gene that was helping with that. Music ability is believed to have a significant genetic component. Resistance to influenza. Again, there was a massive influenza epidemic in 1918. Resistance to that is certainly something that one would not want to eliminate from the population. Cancer is known to have a strong genetic risk, as is are neurodegenerative diseases such as Alzheimer's disease. Now, I'd like to get back to the immunity to plague, because very recently it's been found that there's a genetic variant of a type of molecule that's involved in immune function, which was isolated in people who were immune to AIDS. That is, they could be infected by HIV, but they wouldn't get AIDS. Well, as it turns out, they're mutant for a molecule that is on the surface of immune cells, and although that brings with it a little risk in terms of reduced function of the immune system, it eliminates the ability of the AIDS virus to go beyond a certain point in infecting those cells and results in those people being immune. That's about 2% of the population that are infected with HIV are in fact never develop the AIDS syndrome. The speculation, it's still speculation, has been that the reason that this variant, this allelic variant of this gene, which isn't good really when we have 2 bad copies of it, there's a reduced function of immunity that comes from it, but that the reason is that getting rid of that cell surface molecule also made cells resistant a long time ago to the plague. And for example, in some Finnish populations where there were devastating effects of the bubonic plague, it is believed that their descendants now have fortuitously been made resistant to HIV. And I think this is a great example of how you want to be very, very cautious about what you decide is a bad gene versus a good gene. Under certain circumstances, what you think is a bad gene or a mildly bad gene could be a life-saving variation. In general, it is believed that the genetic variation in the human immune system is responsible for curbing the tides of horrible epidemics like the bubonic plague, like the influenza attack of 1918. And that is that genetic variation that's similar to the sort of tissue incompatibility when people do organ transplants. It is that genetic variation, that intrinsic high degree of genetic variation that is found in various components of the human immune system that act as to limit the spread and magnitude of devastation that are associated with genetic disorders. With contagious diseases. And if we get, in any way, we lower the degree of human genetic diversity, then it is possible, and we homogenize our sort of immune molecules that are present by having elite strains of human beings, which on their own pose certain ethical considerations. But if we do that purely from a medical point of view, and forget about any other types of traits, then what we're potentially doing is limiting the genetic diversity we have in our immune system. And the next time that something like the influenza virus or another virulent virus, which always will be arising, that's something that people who study virology can assure us all, is that there will be new AIDS viruses, new Ebola viruses. These will always be coming up because these viruses are so intrinsically variable in their genetic mechanisms that it is impossible to predict what they will be in the future. And that one day, one of these virulent things will for sure be developed, will develop on its own, and will pose a great health risk. And if we're all genetically the same, instead of having 20% of the world population killed, it could be 90%. And that would be a dramatic catastrophe and would, in principle, be quite avoidable if we don't take the sort of stringent version of eugenics that is possible, where you get rid of the carriers as well as the affected. And I would suggest that you then just get rid of those that you know have the genetic disorder and become blind to those that are simply carriers for the disease, and in this way can ensure the future of the genetic diversity in the human species. And I'd just like to end by saying that although this may seem very science fictional at the moment, that is, these genetic tests don't even exist, and if they did exist, then they would be something that only the rich could apply to use, and it would really not affect the global genetic diversity of humans. Well, that may be true at the very beginning, but when you consider that your genome sequence is probably going to become a routine piece of information associated with you, tests like this are probably going to themselves at some point become quite routine, especially in the Western world. And if you take, for example, what in the press and has been made a great deal of, which is that if you're a woman from Stanford with the proper sort of physical attributes, you can sell your eggs at $50,000 a pop. Given that sort of pressure as it is for the right human genotypes, you can imagine that if misused, this genetic testing scheme might actually be used in a rather systematic way, and I believe to greatly deleterious ends if one doesn't take these considerations into account. Thank you.
There are several things going on here that are undercurrents in our theme today. One is we've showcased some of the academic work in San Diego. One that we've shown that government scientists can contribute. But there's a third thing going on in San Diego that we have to recognize, and that is the contribution of the biotechnology industry, which has come to this town and made it absolutely a world center. So our final speaker, Eric Mather, is from the corporation called Diversa. He's the director of their Biodiversity Access Program. He's been busy constructing environmental gene libraries, working on directed evolution, pathway engineering, and biomolecular discovery programs. He's published extensively and patented a number of his ideas. He's the editor of a journal called Extremophiles. That sounds like something in the sports channel, doesn't it? Extremophiles, and a member of the SETI Life in the Universe Working Group. I think it's very important that we give the microphone now to Eric Mather, because he's going to talk to us about the importance of microbes and their biodiversity. Thank you.
Thank you for the wonderful introduction. I'd like to shift gears rather dramatically here and give you the industrial perspective on biodiversity conservation, and furthermore move away from these charismatic organisms, the elephants, the spotted owls, and plants and reptiles, and even humans, to the real bottom feeders, the blue-collar workers of the environment, the microorganisms. Industry has a stereotype as being the universal or absolute destroyers of biological diversity. But what I'd like to convince you today is the perspective of Diversa, and that is that one of the best ways to preserve an environment is to show that it has value. So let me take you at first about as far as you can get from here to the island of Java in Indonesia. You're on a bioprospecting trip with Diversa Corporation. We're breathlessly watching on as one of our Indonesian colleagues risks his life to take a sample for Diversa. As a geochemist, you might ask, what type of environment is this? Well, it's a boiling mountain. You're in the center of an active volcano with a vigorous outgassing of hydrogen sulfide and carbon dioxide. The surface waters being sampled are about as extreme as can get on Earth, pH 0.0 and 98 degrees Celsius, boiling at that altitude. As a traditional microbiologist, you may say you may assume that no organisms live in this type of environment, pH 0 and 98 degrees. But yet using molecular methods, Diversa scientists are able to detect the signatures of life in these types of pools. Now this is very exciting for a microbial ecologist, but the real question to a biotechnologist is how can you access the genomes and biomolecules produced by organisms living in such environments? Traditional approaches for discovery involve microbiology. A scientist goes out to the environment, collects a soil, water, column, or sediment sample and brings it back to the laboratory to isolate organism in pure culture. They then make fermentation broths of that organism and then attempt to identify novel biomolecules. Well, Diversa's approach is molecular in basis, in foundation. Instead of isolating the microorganisms, we isolate the nucleic acids and thus access that so-called uncultured majority of microorganisms, that 99.9% of genetic diversity that is really not amenable to growth in pure culture. The starting point of all discovery programs is biological diversity. Simply stated, biodiversity encompasses genes, species, and ecosystems and the complex relationship between these components. When you think of biodiversity, it conjures up images as this, of a rainforest with the plants and animals and macroorganisms that make up this environment. But what about the microorganisms? The molecular taxonomic inventories suggest that the vast diversity, the genetic diversity on Earth, is comprised of these microorganisms. Very, very little is known about these organisms. In fact, if you look at phylogenetic trees based on the 16S ribosomal subunit, you see the vast phylogenetic diversity of these organisms, and the metabolic potential of life on Earth is captured in these organisms. At Diversa, we're focused on discovery of enzymes and small molecules from these environments and these organisms. In white, I've highlighted four of these organisms. We've done a complete genome sequence. Pyrolobus here up the top is the highest temperature organism on Earth. It grows up to 113 degrees Celsius and not below 100 degrees. This is a very brand new organism called Nanoarchaeum. Will be coming out in Nature next week from Carl Stetter's work, and this is a obligate parasitic hyperthermophile with less than a 500,000 base pair genome, one of the smallest genomes described. So this untapped microbial resource really has never been looked at using culture-independent methods to really recover their genomes and sift through those genomes for interesting molecules. And so one can go to all different environments, and one of our goals is to access genetic diversity from all environments represented on Earth. So we go to the hydrothermal vents, to caves in Costa Rica, to the rice fields in Indonesia, to hot springs in Costa Rica. And it's becoming increasingly clear that even of the microorganisms being the most diverse group on Earth, yet the vast majority of these organisms are not even amenable to cultivation. In fact, if you look at open ocean water, less than 1/10% of the organisms present are amenable to cultivation. And one of the most studied environments on Earth, activated sludge from bioreactors, a full 85% of the organisms present in these industrial bioreactors have not been characterized. So clearly there's an untapped resource for discovery in these uncultured organisms. And so gone are the days that you can send your scientists, industry can send their scientists out to on vacation and collect samples in their suitcase of soil and water and bring them back in commercialized genetic material. The Conservation of Biological Diversity program, now ratified by 174 nations, gives each nation sovereign rights to their genetic and biochemical resources. So in order to really collect a sample from a country and commercialize that sample, one has to have legal rights from that country. And at Diversa, we've gone out of our way to set up these bioprospecting programs consistent with the Convention of Biological Diversity, calling for conservation of the biological resource, sustainable use of its components, and most importantly, equitable benefit sharing. So some of the benefits from the sampling of these environments goes back to conservation of that particular environment. And so we've set up these bioprospecting frameworks at Diversa, and I want to contrast this with the biopiracy, which is a term often kind of confused with bioprospecting. Bioprospecting is the exploration of biological diversity for commercially valuable resources, and it results in the protection of wild lands, whereas biopiracy is the unauthorized and uncompensated collection of samples from such environments. And so it's very important in today, moving forward more so, that we have legal rights, and companies, when they're working with biological diversity, are working within the system, within the government, and not just getting a permit from a state lab or from someone owning the property. You need to work with the government in terms of biodiversity access. And this framework we developed, and this is unique to Diversa, is very dependent on the country. We work everywhere from Indonesia to Bermuda to Costa Rica to Russia. And in each case, we build a framework of both monetary and non-monetary benefits. Of course, we have the milestone and royalties associated with products developed, and collection costs and help in laboratory funding. But more important are the non-monetary benefits involving capacity building, teaching the young scientists. We have training workshops. For example, South Africans come to our laboratory, and folks from Ghana come and get trained in technology development. Not only technology related to the work they do with Diversa, but also technology just to move the country to the next generation and get out of this emerging status. And I think the real future is in the young people. And so training these young people in molecular technology really will advance the systems very far. We also have equipment and supply donations and grants and publications together, and so it's a framework customized for each environment and each different institution we work with. And obviously a big problem on Earth today is deforestation, and the numbers are just amazing. 130,000 kilometers per year of rainforests that are destroyed. And these are strip miners and cattle grazers who are looking for short-term benefits to destroying their land. And through working with InBio Corporation in Costa Rica, the model is that instead of having these strip miners and cattle grazers destroy their native endemic indigenous environment, we train them as parataxonomists to actually collect samples for Diversa. And then instead of destroying the environment, they become stewards of that environment. So it's a way to show that the environment has value. And it's a very powerful paradigm shift in thinking. And to take that even a step further, you see here, this is a French company is very interested in perfume aerosols. And so these Costa Rican scientists are capturing the gases released from these arboreal plants, the different chemicals releasement plants and capturing them and actually selling the air to the French companies. So this is as sustainable use as you can get. It's very non-invasive. And these types of paradigms, using these for moving forward in terms of conservation efforts, show value. And suddenly this rainforest, which could produce short-term benefits for cattle grazing, for hamburgers, for a company, now suddenly becomes a valuable resource for these chemicals that could be used in perfumes. So this is the paradigm that we're trying to move toward here. And so we collect this diversity from all ecosystems on Earth and generate highly representative environmental libraries. I'm not going to get too much into that today, but suffice it to say we have very high throughput screening systems. We have fax-based flow cytometry systems that can screen 50,000 clones per second. We have these new methods we've developed at Diversa called Gigamatrix, and this is a footprint of a 96 well, one well of a 96 well plate. These plates we have 100,000 wells in one plate, the size of a human hair, holding about 250 nanoliters. And so our discovery efforts, when working with diversity, suddenly the numbers become very, very big, and you can't just, it's not like looking at one organism. When you're dealing with clones from the environment, suddenly the numbers are huge, and you have to develop very high throughput systems to accommodate these methods. And now let me give you an example of the type of work we do in our science. So here we went to a dusty field not far from Owens Valley in Central California, and isolated the DNA from that soil sample. And we found out by using molecular phylogeny techniques, we have over 5,000 genomes present in that organism. And we made a library, a very large library, about 10 to the 7th clones. Put that in perspective, if you have 10,000 clones on a plate, that's close to 10,000 plates in order to screen. So very, very complex diversity, 5,000 unique genomes in that library. And then we screened that library for esterase activity, an important enzyme used for chiral resolution of pharmaceutical and agricultural intermediates. It turns out we found from only a small sampling of that library over 120 unique esterases, and in the whole public domain there's less than 200 esterases. And it's a very interesting story, and I'll show you a little more about this. These are the phylogenetic trees from that one sample. In yellow are the signatures of life, the 16S subunit that we detected. And it almost reads like a phylogenetic tree. You see the gamma and alpha proteobacteria, green sulfur bacteria, the planktomyces, the gram-positive IgC. Huge numbers of organisms found, and even these thermotogales. And there's an organism here that's related to the Antarctic Cranarchium that Ed DeLong discovered, found in the deserts of California. So this is really exciting. We find numbers of just tremendous organisms. But what's exciting then is we based on phylogeny, we see all this diversity based on 16S subunit. We then say, well, is there protein diversity? And here is the protein tree of the esterases found in that one sample. Now, esterases were thought to be convergent evolution because there's no homology besides one catalytic triad. But yet in this one sample, we started seeing homologies and families fall out. And now we have 22 unique families of this one class of proteins, industrially valuable proteins, from one sample. And if you go to other samples, you find whole new classes. And so in terms of microbial diversity and the amount of genetic diversity present in those samples, it's unfathomable. It's interesting, it's a nice party trick, but is it valuable? And that's really the question what I'm here for today. And this shows you an example here of a resolution of an enantiomer mixture of molecules in the preparation of agricultural intermediate. No enzymes on the commercial marketplace had activity on this enzyme. We gave them 50 samples of these enzymes of ours, and these 5 had activity on the substrate. This first one had 100% conversion to from the product substrate to the product, but no selectivity, enantioselectivity. Whereas this one right here had absolute 100% possible theoretical conversion to the proper enantiomer. So having these large collections of enzymes captured from diversity are valuable, and that translates back to going to the environment where they came from, and suddenly that's valuable. This dusty desert road in California suddenly becomes a very important resource. And to show you a little more about the discovery program here, these are 2 important classes of enzymes, nitrolases and haloalkane dehalogenases. Again, these are very important in chiral resolutions in production of pharmaceuticals, and haloalkane dehalogenases are used in remediation. This is the collection, the world collection of these enzymes, and there's 9 nitrolases and several, I think 4 dehalogenases. After our discovery programs, we fleshed these out with huge numbers of proteins. Now they're related, they're not identical, they're all different amino acid sequences, but related and have similar functionality but different. So to find the right enzymes for the right activity, it's good to have a large collection. We have a whole program on molecular evolution and gene blending that I'm not going to speak about, but having these large numbers of starting points sets you up perfectly for gene blending experiments to really customize enzymes and molecules for the proper application. And these are some of the types of applications we work with. Oxyonitrilases, polymerases, dehalogenases, hydantoinases, and there's a whole number of intermediate products and a lot of interest from industry. And taking again this back to the beginning, the biodiversity, you're collecting a soil sample from the Volga River in Russia, which is a pretty obscure contaminated site, but it has very valuable enzymes. And suddenly this one site in Volga River could become important, and we can bring royalties back and put money back into that site for preservation. This gives you a little more resolution of one of the programs we have going, and we've developed an industrial enzymatic route to produce Lipitor, which is one of the most prescribed drugs on the market today. And so these enzymes truly have huge value, and accessing diversity results in this value. So suddenly putting these pieces together, conservation biology and value from industry, you can start developing a paradigm of conserving the environment for discovery. This is an example of, I'll close here with a few products that we have coming from diversity. And this is an alpha amylase for making high fructose corn syrup. In industry, the reaction is, you want it at 4.5 pH. The current enzymes here are shown, and an improvement from in 1998 what raised it here, but still very poor activity. And going to natural diversity, we find tremendous benefits. And now this is a very large market, and it's going to be released at the end of this year for an enzyme from diversity that's going to result in significant change in the sugar corn wet milling industry. Another product we have developed is a phytase for animal feeds, and this is phytase is an enzyme that takes phosphate off phytic acid. And this is phytic acid here, and 80% of phosphate is tied up in legumes as phytic acid and is not available to the animal. So what happens is when the animal, chickens and pigs, monogastric animals eat these grains, the phytase is excreted into the environment. First of all, they get no value from it, so you have to add phosphate and metals ions, but the phytic acid is excreted into the environment and then digested by bacteria in the environment and creates phosphate pollution, which results in cyanobacterial blooms and red tides. And so suddenly, this is a pollutant. Adding this enzyme, this benign enzyme, to animal feed can increase efficiency of the feed and reduce pollution. And the long-term goal, as we're hearing earlier today, is to put this gene into the plant itself, into barley. So there's really no cost to the grower or to the animal feed, and you suddenly have a process that improves the efficiency of the grain conversion in animal and also reduces the pollution. And I've talked a little bit about these industrial and agricultural sectors. So we have a large collaboration and joint venture with Dow Chemical here in San Diego. We're working on a joint venture called Innovase, where we're focused on industrial enzymes like this amylase. And we have another collaboration with Syngenta, which is now the largest agricultural company, merged of Zeneca and Novartis. And there we have a joint venture called Zymetrix, focused on animal feed enzymes, and that's in the agriculture and ag product processing area. We also have two other programs, one with GlaxoSmithKline on small molecule discovery from the environment, and then another program on protein therapeutics and antibodies. So it's a whole platform, all with its foundation on biological diversity. And it's, I can't overstress the power and value of biodiversity, and unlike the paradigm and the stereotype of industry being, the ultimate destroyers of diversity, that paradigm's changing. And I think companies such as ours are really moving toward really helping and doing our best to preserve the environment. And let me close with just a few more words about Diversa Corporation. We were founded in 1994. Our head of research and chief executive officer is Jay Short. We had a very successful IPO in February 2000 and have about 300 employees here in San Diego. You might see our building as you go by the merge. I'd like to also have a quote here from E.O. Wilson, where he said, "If I could do it all over again and relive my vision in the 21st century, I would be a microbial ecologist. 10 billion bacteria live in a gram of ordinary soil, a mere pinch between the thumb and forefinger. They represent thousands of species, almost none of which are known to science." And that was from The Naturalist.
Thank you very much. If you would join with me and thank the other panelists for their wonderful talks, I think that would be the next thing to do. Thank you very much. I think I would like to ensure that we give you a chance to ask questions. That was the purpose. And then, of course, there'll be more opportunity during lunch for private conversations.
Suppose this question is directed toward Dr. Woodruff and Dr. Fisher. One of you or both of you had made mention of the habitat conservation plan. There's two plans underway in San Diego County to provide habitat linkages through urbanized areas. That whole process, as we know, is really slow going. And what real prospects do those hold to save or protect individual species? And is this kind of work being modeled anywhere else in the country, or is San Diego really the only one that is looking at such a coordinated focus on habitat linkages?
I'd like to start by setting up Robert to answer the question, because he is the expert, I think. When I said we are a national test bed for responses to the biodiversity crisis, I really meant that. The coming together of the San Diego area governments to produce the MSCP in its infancy is a work in progress, is an experiment, is something that hasn't been done, and so there are few other examples. And so the federal government is interested in making San Diego a test for processes that could be adopted elsewhere, because the problems are clearly generic. Is it a work in progress? I'll let Robert tell you how frustrating it is to watch it, even when the scientists provide the data.
Yeah, I think at the scale that we're talking about, and I mean, we're, like I said, in the middle of this biodiversity hotspot. So to plan for biodiversity when you've got 16 million people down here, you know, and 5,000 square miles and billions of dollars worth of land, it's a very difficult situation. It's very much different than if we were doing this in South Dakota or Iowa or other areas where you might be able to do some other kinds of trade-offs. And so it's not just that we're attempting to do something that's, first we're attempting to do it in one of the most complex situations, environments. And so I think there's not many options. I mean, the options are we continue to develop as we have been, which is why we're in a situation where we're in right now through land use change, or we work as a group with industry, with development, with the local governments, and try to come up with a solution that works as best as it can for local biodiversity, but also it keeps everybody at the table. I mean, because you can't. There's so much money and so much of our national economics is linked to Southern California that you can't just stop growth in Southern California. No one could do that. And so that said, you have to figure out a way to work with that. And if we can solve that here, at least to some extent, I mean, we're not going to get jaguars back. We're going to maybe keep mountain lions. We're not going to have grizzly bears in Southern California in the future. But if we can identify what those goals are for the different plans and for those different landscapes, then we can have some functioning ecosystem and maintain biodiversity and the processes associated with biodiversity, the evolutionary processes, and do that in this complex landscape as we kind of understand also the effects of edge and light and all roads, and you know, we can then try to solve those by linking with industry. I was at this light conference, and all the light engineers are there trying to figure out how do you darken the skies in Southern California, and so it's going to take a lot of different progressive links to solve some of these problems. But I think that I don't think that there's any place that's like this with this level of complexity, you know, that you could hold up as an example. I mean, Florida is doing a lot, but nothing's comparable to this with the amount of investments and stuff.
Can we have a second question, please, from the second row?
I had a question about the genetic crop diversity, and I loved your goal. I have lots of questions, but there is lunch. Increasing crop productivity and preserving the natural ecosystems. I think it's a marvelous goal. HCPs and CCPs are sort of Response to Endangered Species Act, but what would you say are the required mechanisms, or what should the required mechanisms be, especially for this crop productivity, in the crop productivity arena, to preserve preserve the natural ecosystems, and who should bear that responsibility? Who's bearing it now?
Well, as I showed you on the world map, there are areas where productivity could be significantly increased, and those are also the areas where the least research is being done. So, as you know, I didn't talk about the Green Revolution, but the Green Revolution greatly increased agricultural productivity in parts of Latin America, in a large part of Asia also, but not at all in Africa. Africa is ecologically extremely diverse and has crops that the Green Revolution did not address or did not utilize. For example, the Africans eat cassava, they eat millet, they eat cowpeas, and they eat those crops because those crops are adapted to those ecosystems. So essentially no research has been done on those crops, no breeding, no improvement, even classical types of plant breeding. So an enormous amount of research needs to be done to increase productivity in those areas. Now, who should do this? Well, and who should fund it? Well, the Africans clearly don't have the money to do this. So there are some international research institutes that are based in Africa, for example, and they do work and must work with the so-called natural agricultural research systems. So, and we now understand that most of the solutions to this problem have to come not top-down but bottom-up. So the scientists in the national agricultural research system have to work with the farmers on the one hand and with the international institutes on the other hand. A great deal more money is needed from the developed world to bring this about. There's no doubt about that. And the United States has always been extremely niggardly in its willingness to fund development assistance. As you know, finally Bush is coming through with what will hopefully be a 50% increase, but we are at the bottom. Per capita, we are at the bottom of all the industrialized nations in our support for such activities.
And I guess as a follow-on question, if I may, can we make a stronger linkage between that research to increase crop productivity and to protect natural ecosystems? Because I think right now there's a big disconnect.
Well, it won't happen unless we increase agricultural productivity. The pressure to produce more food is there. As you see by the deforestation, for example, this is part of the enormous pressure there is that as people get richer, they want to eat more meat. So they don't want to eat beans and rice. They want to eat steaks and hamburgers. So it certainly won't happen. That is, those ecosystems won't be preserved unless agricultural productivity increases. Now, clearly, the various nations where this is happening have to take stronger regulatory action or stronger enforcement action of the laws they do have. For example, Brazil probably has very good environmental laws, but the problem is enforcing them. I'm not an expert in this field, but I'm sure David could talk about this.
Another question from the front, yes.
My question is actually for the whole panel, because I sense that there are some similarities, whether it's animals, whether it's plants, whether it's the human genome or microbes, that we don't seem to be very well prepared to become the caretakers of diversity. We don't know very much in the whole scope of things. So how do we feel like, how are we prepared to take on that role? Obviously there's work in that direction from all of you, but...
Well, it's not just taking on a new role, it's a totally different way of approaching nature, and the sudden realization, by sudden I mean in my lifetime, and I'm not that old, that we are the only species responsible for the predicament that we're in, and we have a sole responsibility for stewardship. There's no other species that's going to step up to the plate. For me, one of the interesting challenges, and I'd like Eric and others to comment on this, is that just as the scientists aren't equal to the challenge that's been dumped on us, suddenly we've been asked to play God. Society isn't yet prepared to step up and deal with the ethical issues and fallout that come along with the science we wish to apply, that we have to apply. Ethan touched on the ethics of designer babies. Obviously, if Martin fails to feed the 9 billion people, we'll have to turn to Eric for frankenfoods made of microbial compounds to feed people. Society isn't prepared to deal with that, and unfortunately, the press, although the press carries more and more environmental stories, they often carry them in ways that do not help people move through these difficult ethical questions to the other side where we have to go. So we still see stories played for shock value rather than stories of the world has changed, the rules have changed, let's move ahead. Eric, would you like to comment?
Sure. First off, I think that work by Dan Jansen and others in this all taxonomic biological inventory programs where they start categorizing and inventorying all the organisms on Earth. I mean, this is a good starting point, and it's more straightforward with some of the macrofauna, but as we heard this morning, even some of these elephant populations, people don't know who's there. So having a good broad understanding of the taxonomic inventory is a real good starting point. I think, and in terms of microbes, we're doing this and setting up these microbial diversity centers in different extreme environments. 12 of our centers are biodiversity hotspots. 12 of the 25, and so in those centers, we're one of the capacity building areas we work with the institution there. Our collaborating institution is molecular taxonomic inventories of the microorganisms. We're moving beyond that, for example, in the coral sponges and the metazoans and off of Costa Rica and off of Bermuda, where we're actually beginning to do taxonomic inventory, molecular taxonomic inventories of the metazoans, which really no one's ever done. So having this baseline, I think, is a starting, but I think what you're saying is very true, that there's going to be problems here, and we need to really address it as a broad, sophisticated group.
Other questions here, please? Thank you.
I do, actually, for Eric. My question is, in San Diego, has Diversa done collecting in San Diego, and have you noticed that there's been any effect of your collecting on the valuation of lands preserved in the MSCP by our preserve managers and so on? Because I know personally that we have very little money to manage these lands, and I'd like to know if you're having any local effect.
Well, that's interesting. I mean, at the end of the day, it's the Convention of Biological Diversity that governs our relationships, and this, again, this convention is not ratified yet by the United States. We're in the elite crowd of Somalia and Afghanistan and a few other countries not having ratified this convention. But with that said, it's very difficult to work in the United States because of stewardship of land. We work in Yellowstone. It's pretty clear the stewardship there. But if you go, for example, to the side of the road here on I-5, is it the Bureau of Land Management? Is it the Coastal Commission? Is it the Fish and Game? There's so many stewards of land, and nowhere in any regs is there anything about commercialization of genetic resources. So it becomes very difficult, even as an education process in working with any institution anywhere. And so our primary efforts have been with biodiversity centers around the world, but we have been talking to Ollie Ryder at the San Diego Zoo and the CRESS, Center for Reproduction of Endangered Species, accessing and working there. We talked a little bit offline this morning. One project we had is looking at African black rhinos in Kenya. We're looking at the microbial flora in their dung and comparing that to the microbial flora in the dung of the captive species, because they're having trouble in captivity, and there may be some signatures that come out of looking at the differences in microflora in the dung based on rhino chow versus the standard endemic diet. But we haven't really worked too much in San Diego besides in private land, but we'd be very open to.
Well, Robert Fisher, can you have any insight into that? If Diversa wants to collect on federal land, are they going to have huge regulatory problems?
I mean, I think they just, I think, like you said, with the national parks, with just Yellowstone, it's pretty obvious where the boundaries are. And I think a lot of our lands, with MSCP, I mean, they're still transitioning. So I think if they had could identify places, then they could probably figure out, who is the owner, and then a lot of these agencies haven't even thought about it. So I mean,
that's the problem.
How do you get a special use permit from refuges to do the stuff that they're talking about? If you're just collecting the dirt sample, that's one thing. But if you're then going to patent some product from that dirt sample, that's a different set. I think it's something that could be resolvable. I just don't think it's beyond where people are thinking right now.
We've had a lot of activity. Didn't talk about Yellowstone too much, but we were the first of its kind created with Yellowstone, to showing that we can preserve and actually help bring revenues into conservation of Yellowstone by showing there's value there. So taking literally teaspoons of sediment from obscure hot springs in the backcountry and commercializing products that result in value that can go back to that environment is a really powerful concept. But even that has been frayed with the loss. There was a lawsuit against National Park for them entering into the crater with us. It has since been upheld, and even so, we as a company want to make sure it's held up absolutely to light before we do anything there. So we suspended all of our discovery activities on those samples, even though the courts have said it's okay, but it's still under environmental impact review, and there's so many levels of problems here. And then also on top of that, you have some of these groups that want to get on a soapbox and say that this should be stopped, and some groups want to stop all cloning of genes, and they can use these sort of things as case studies. And so it's very complex, I think.
As in all big societal changes, we find the science isn't equal to it yet. Human institutions weren't designed to even address these problems in a sensible way. And just as medicine is sometimes thwarted by traditional beliefs or religious strictures, that's the state we're in today. Another question from the front, please.
So one question along the lines of biodiversity here in San Diego. Have there been studies of whether or to what degree biodiversity in wetlands and lagoon areas have possibly fallen because of a decline in tidal flushing of those lagoon areas?
Yeah, I mean, I think there's a pretty clear link there. And also because of access issues. I mean, the work that's been done by Scripps showing that, just people going to beaches, and taking home the fauna with them. And that there's terrestrial analogs to that with reptiles, where people take home the horned lizards and snakes and stuff. So I mean, I think that's another social element to this biodiversity is understanding, that what's a park and what's not a park. And
have those studies been done on a microbial level, though, to look at that level of diversity in lagoons that have been lagoon and wetland areas that have been cut off from ocean flushing?
I can answer that population has changed dramatically. Even if you take a handful of soil, put in a baggie, by the time you get to the lab, it's changed. So populations change dramatically, and I would imagine and suspect that any kind of contamination or any kind of environmental impact will dramatically change the flora.
I have a question here.
Our habitat plans, the preserves that are resulting, tend to be linear, highly fragmented with a lot of edge. And I think what I've heard today is that the allele frequency and even the organismal diversity is going to decrease no matter what we do because of the amount of land in those preserves. So I'm sort of curious, if military lands, Camp Pendleton and Miramar and Otay Mountain are the big land masses that I know about, but if military lands get exempted from the Endangered Species Act, what's the long-term consequences to the preserve being able to support species without those large land masses feeding into them?
Robert?
For one, I mean, as you know, that's just a political ploy. The same thing when the Border Patrol did with Homeland Security. I mean, Homeland Security is going to be a big issue this next year. That's where federal money is going to go. And Homeland Security is going to push to be exempted probably the same way the military is. And I think that there's enough concern about our own resources at home that in Congress that a lot of those things aren't, you know, it's a ploy by the agency, but it's not going to get approved by Congress and not get buy-in by the populace.
But if it were, what would be the consequences to the species and what is going to be our preserve?
Yeah, I mean, no idea, because it just doesn't seem like it's a possibility. Yeah.
You're going to lose species. You can predict which ones will go first to some extent, and you can predict that the ones that stay will depend on you for help to survive in the long term. I asked a government official to answer your question because I believe, I'm not positive, that I was basically invited off San Clemente Island as a result of overstepping my role as a geneticist in that particular recovery program. Stewardship of the land is not a priority for the Department of Defense units. Until it became a priority, their other activities will come first. So again, we have an institution that we're now asking to do something that it was never designed to do, and society and the politicians who represent society have to change the mission of the institutions if they want it to serve these other functions. I'd like one more question, and then I think we'll have lunch. Is there one more out there, please? Yes, at the back. Thank you.
Actually, a very brief comment, if I may, to clarify about the regional habitat plan. I think it's important, and this is neither a criticism nor an endorsement, but I think it's important to point out the fact that the multiple species plan doesn't protect more habitat than conventional administration of the Endangered Species Act. In theory, it actually protects less habitat, but it's habitat in the appropriate location. So I think that that's an important point, and also the fact that it is negotiated in a political context, and so to a large extent it's shaped by the political landscape of our region. There are several hundred habitat conservation plans being developed, several of which are regional habitat plans like ours, and we are being looked at as a model. So it's an important discussion to have. Thank you.
I'd like to thank everybody who came to this morning's presentations and for making the discussion so interesting. I invite you all now to do what we do best, which is to break up into small groups and have useful conversations over lunch.